Good morning, everyone. We're going to be starting here in just a few more minutes. And we hope that we can send the correct link to everyone who registered to join. Hello and welcome, everyone. We're going to be starting in just a few more minutes. And I think there was a mistake with the link that was sent out. So hopefully everyone will join the correct meeting this time. And today we're going to be talking about bioinformatics for infectious diseases. So please give it just a few more minutes as everyone is joining, and we'll be starting soon. All right, hello and welcome to this webinar on bioinformatics for infectious diseases. We're going to be starting here in just a second. So thank you for joining today. And we're going to be discussing the applications of bioinformatics to the study of infectious diseases. Anyone who's joining us not on Zoom, we do have an issue. I think someone sent out the wrong link to join this session on Zoom. So if you are participating from YouTube, that's also great to have you all here. So bioinformatics for infectious diseases will be the topic of today's session. But before we actually start the session, I wanted to talk a little bit about what we do in the Omics Logic training in general. And that will kind of give the context for what we will be talking about today. So Omics Logic is essentially a program that combines the logic of data science in application to biological data with a community that is passionate about developing research projects using bioinformatics tools and applications of bioinformatics to a variety of topics. And so we believe that the students, clinicians, faculty of all backgrounds need to be enabled with the skills and the tools that are needed to process large data sets. So as we go through the first several steps, I want to introduce you to some of these resources that make this possible. And after we go through this first part, I will be talking about the topics of the Bioinformatics for Infectious Disease program. And as we will try a few things with the hands-on exercises that we'll do today. So hopefully you can also start by registering on the platform that we will use before we start this session. Okay, if you're having trouble hearing, please rejoin again. And Mohit and Sonalika, am I audible? Can you hear everything fine?
Yes, sir, you're audible.
All right, great, thank you. Okay, so before we begin, let me just go through some of the resources that we will be using today. The first one is learnomicslogic.com. Now, some of you might already be registered on this portal. So if you already are registered, please make sure that you're logged in and you have your password ready, because we're going to be doing a few examples on our big data analysis platform to kind of give you a sense of what kind of exercises we will be doing. If you could please post the link to the Learn portal in the chat, anyone that wants to register. So learn.omicslogic.com, and the way to register, and just go to the site, is to go right here to the login, and then you can either log in with your email and password or create an account, or you can also use one of the social media portals platforms here to create an account. Thank you, Mohit. So once you're logged in, you will see a number of resources, and we will be talking about some of these resources today. So to gain free access to a lot of these resources, all you need is the registration on the portal. So some of these resources are going to be useful in the program as well as in today's session. And after today's session, you can explore them a little bit more. So some of the things that we'll talk about in these resources are the terminology that we'll be using today, the logic of analysis, building different pipelines for bioinformatics processing and analysis of data. What are some of the technical skills that you will be needing, knowing how to visualize data and explore that data using R and Python. And then also you will see how you can evaluate your understanding of some of the topics that we will be covering by covering project applications of bioinformatics methods or quizzes for self-evaluation. Now, importantly, we think that these elements really complete a successful journey for anyone coming from a very introductory background. So you might not know anything about bioinformatics before joining this program, or you might already be an engineer or a biologist. And the way that we view these two parts of bioinformatics, the biology and the data, is that really they merge together as we start speaking about some of the applications of bioinformatics. And the only way to explore the applications of bioinformatics is to really focus on examples, on projects. And so what Omics Logic portal does is it offers a variety of specialization tracks, such as oncology, infectious diseases, neuroscience, and agriculture, for anyone who's interested in these topics to appreciate the role data science and bioinformatics can help in the research areas, and also in translational research, how to develop new applications for medical purposes, pharmaceutical, agriculture, and others. And so what the portal offers is the two separate tracks on biology, on data science, using curriculum that was evaluated by experts in these fields and providing resources that could be leveraged for an effective program. Now, besides the program that we are using, that we are discussing today, we have several other tracks that you can benefit from. So you can look at Introduction to Bioinformatics to kind of get started and understand what it's all about. You can learn specifically about genomic data analysis and application to infectious diseases, oncology, or neuroscience, transcriptomic data analysis for immunology studies or for understanding of gene expression in complex diseases, metagenomic analysis of data from the microbiome, where you sample environmental samples and analyze their composition, epigenomics, the regulation of non-genomic variation in response to different diseases or environmental changes, as well as a training program on data science. And that program is specifically for those that would like to get practical hands-on skills for data visualization, data wrangling, statistical analysis, as well as machine learning and artificial intelligence. And so those programs will be offered. And at the end of today's session, we will hear from Dr. Mohit Mazumdar, who will describe what are some of the upcoming programs that we have on a rolling basis. Together, these provide a complete roadmap for data science in application to biological data or biomedical research. And so you can see how finding data sets, identifying patterns through visualization, performing statistical analysis, and understanding the research methods that you might read about or you might be familiar with, as well as gaining biological insights into these data sets, providing with evidence-based or data-driven hypotheses. So that's really the focus of this portal. And hopefully by now, all of you have had a chance to create an account. Now, who benefits from such training? We have a few examples here. As you can see, this could include faculty, researchers, students. We serve a community of participants from all around the world, including India, Africa, the US, and Europe. And so we are excited to kind of continue and develop and build this community that comes from very diverse backgrounds, but with a common interest of application of bioinformatics to a variety of biomedical and biological challenges. So to enhance this experience, we also welcome you to join our group on LinkedIn. We have a group of professionals that offer opportunities for internship and jobs, career building. So if you're interested in the application of bioinformatics to a variety of biomedical challenges and you're looking for a job or you're looking for an internship, we welcome you to join this community of experts and students alike so that you can learn about different training programs, internships, jobs, and understand how experts have gotten to where they are today. So to join this LinkedIn group, if Mohit, you could share the link again for the LinkedIn group. And we also have a group on Facebook. Also, for those of you that use Facebook, you can also join that social media platform where we do the same. We post insightful resources as well as opportunities for internships, mentorship, job applications, and our participants that complete our training are able to share some of their projects as a result of the research fellowship that they conduct with our company. So besides the asynchronous resources that we have covered today, the program that we're going to be discussing today, the program is called Bioinformatics for Infectious Diseases, will consist of several different elements that work together to provide an experience, an immersive experience for anybody that would like to learn about the applications of bioinformatics for infectious diseases. So this will include interactive sessions like we're having today. It will point you to the right kind of self-guided study materials that you can use in between sessions to cover the topics that we don't have enough time to cover, as well as some practical assignments. So you'll actually be given curated data sets. You'll be able to perform analysis on our big data analysis platform that we'll introduce you to today, as well as some coding exercises so that you can explore, visualize, and analyze this data for biological interpretation. But for now, let's take a little pause. I think that's enough about us and what we do. And before we move on, I wanted to ask you, what is your background and where are you coming to this session from? So the first question that I have, what is your current level of education? So I have a few answers here on the screen. High school, undergraduate degree like a bachelor's, graduate degree like a master's degree, or postgraduate, PhD, postdoc, or faculty. So if you could put your answers in the chat. For anyone joining just now, we are speaking about bioinformatics for infectious diseases, but I wanted to get to know the audience as well. So if you could please put the answers in the chat. Thank you, Carrie. Okay, so 1D would be postgraduate, and then Diksha is answering undergraduate degree. Okay, Kavya, is saying D. So a lot of postgraduate, undergraduate degree for Shayani. All right, great. Thank you. Vijay is D. And Ali is saying graduate degree. All right, so welcome for all of you that are joining now. So great to have you here. And Louis is saying D, which is postgraduate. All right, excellent. All right, great, thank you. And the other question is about what kind of drove you, right? What is the motivation for you joining this session? So again, this is number 2. So that's why I asked 1A, 1B, 1C, and then, this is number 2, 2A, 2B, 2C, and 2D. So the first option is to learn about bioinformatics. Maybe you're coming with minimal background in bioinformatics and you just want to explore and understand a little bit more about it. Number 2, to get familiar with infectious diseases. Maybe you have more experience in bioinformatics and application of something else, but you would like to understand the applications to infectious diseases. For overview of bioinformatics and virology, so infectious diseases includes bacteria, pathogens from parasites, bacteria, fungi. Okay, thank you, Ali. And then D, to apply bioinformatics to a research project. So maybe you already have a project in mind, maybe you have explored some data sets and you would like to have some mentor guidance and a better understanding of how to properly apply different types of bioinformatics methods to a project of your own. Potentially you're already a researcher trying to use those skills. All right, great. So I see a lot of you are putting the D for question number 2, and then also C, an overview of bioinformatics of virology. Thank you. Okay, so for those of you that are interested in that last part of it, to understand and to apply different types of bioinformatics methods to projects, to data sets from infectious diseases, that's exactly what we want to cover in today's session. So I'll give you a little bit of an overview of bioinformatics and application to different types of pathogens. I will then introduce you to some of the methods and data sets that we will be using during this program. And I will pause in between those different sections so that you can ask any questions that we can pause and discuss in today's session, answer any of your questions, and also help you envision how this program can help you achieve some of these goals. So I will not be the only one speaking both in today's session, but also throughout the program. We actually have a number of experienced mentors that will be guiding and assisting you throughout this program. And that includes Dr. Harpreet Kaur, who is a Omics Logic bioinformatics trainer and has a PhD in bioinformatics, particularly with a specialization in genomics and oncology. As well as Dr. Mohit Mazumdar, who's also here in the chat, and he is an expert in chemoinformatics, as well as the application or the relationship between sequence and structure. And I'm sure that he will mention a little bit about his background towards the end of today's session. And so my name is Ilya Brodsky. I'm one of the co-founders and the CEO of Pine Biotech, but I'm involved in every aspect of what our company does. And I'm particularly excited about this program because I think it's on everyone's mind today. Even people that used to be interested in oncology and neuroscience are now seeing the impact of infectious diseases on so many areas that we have been interested in before. And I think that it's impossible to kind of overlook the significance of what's going on. Not only are we living in the midst of a pandemic, we always hear in the news how there's new development. And so because of these developments, it's important to understand what we can do with the type of data that is available in such great quantities on the public domain, and how we can actually contribute by analyzing such data to extract meaningful insights from some of these data sets. So what can they tell us essentially? So this program called Bioinformatics for Infectious Diseases is going to focus on the relevant tools, methods, data repositories, and provide some examples from curated data sets where we can take sample data and analyze it together during these interactive sessions point you to some project examples on the Learn Omics Logic portal, where you can register for free, and we'll be utilizing some of those examples and tutorials during the session, so that you can master some key elements of analysis and visualization tools, and hopefully apply some of these methods to a project of your own. So the objective of this program, and what we sincerely hope for everyone that joins this program, is that you will start and learn about something that is relevant to a research project that you would like to develop. Now, this program is going to last for a little bit longer than just the previous program that we completed not so long ago. That program was exclusively focused on the COVID-19 variants. There's some overlap between that program and this program, but we hope to widen the perspective so that we can look at sequence variation, evolutionary and phylogenetic analysis like we've done before, talk about functionality and protein structure using several different examples, and then also look at the transcriptomic data analysis so that we understand the host responses and potentially some of the immunology of these infectious diseases. So let me, for those of you that are joining with minimal background in virology or infectious diseases, kind of start from a little bit of a beginning. What are infectious diseases and how are they different from non-infectious diseases? So infectious diseases spread by pathogenic microorganisms, and those can include bacteria, viruses, parasites, and fungi. And you can see some of the examples here on the left side. So flu, HIV, malaria, and some of these are very different types of diseases. So they affect different tissues, they affect different types of cells, they have very different mechanisms of infection. But we call them infectious diseases because there are several characteristics about them that are unique. So for example, transmission is an important factor. The way that they replicate is going to be an important factor. And this is going to be different from non-infectious diseases that are not caused by a pathogen. So typically they are considered to be caused by environmental factors, nutrition deficiencies, lifestyle choices, and genetic inheritance. And when we speak about this approach of studying infectious diseases, some of the things that we will be focusing on are replication or colonization, cell death. So how does the pathogen induce the process of deterioration and death, as well as release of toxic, and as a result of that, how we can understand organ and tissue damage. So these are some of the important factors. And surprisingly, in maybe the past 10 or 20 years, the researchers have discovered that even though we might not consider some diseases to be caused by pathogens, some of the conventional diseases that we think of as non-infectious, like cancer and neurodegenerative diseases, might be associated with some of these factors. And one of the primary reasons for that is actually related to the way that our body responds to infectious diseases. And so to talk a little bit about the response, you can see here that, for example, bacteria, and you can see that it is being, the response is the ability of the body to kind of intercept, right? I don't know how to properly say that, but to intervene. And so when we speak about infectious diseases, we also have to consider the immune response to those infectious diseases, where this rapid ability of the pathogen to replicate and colonize different tissues is going to be important for, okay. Sorry, 1 second. It looks like somebody signed me out of this. Okay, so, okay, Mohit and everyone else, if you're trying to use my account, please stop that. I think I need it for my presentation.
Okay, Ilya. So actually the meeting with, there's another meeting with African team about machine learning. So
Well, maybe tell them to use your account or something else because we do need this for now.
sure. Okay. This is mine actually. So yeah.
Okay. Okay, so as a result, we need to consider the history of interventions to help address the spread of infectious diseases. And over the course of the years, there have been several important interventions at the public health level that have been very successful at reducing the load from infectious diseases and mortality. So you can see here the rate of infection and the years, and some of the important interventions that have been developed over the year, like the health departments and the public health component. Then you can see the first continuous municipal use of chlorine in water in the United States. After pandemics, especially, these novel interventions have been applied in a much greater way. And then you can see how it helped intervene with transmission of important infections, the use of penicillin and the development of antibiotics, as well as not so long ago, if you think about it, in the 1950s, the introduction of effective vaccination. And so the types of challenges that we are facing today, the reason why they are so unexpected is that we have a fairly successful history of applying our understanding of infectious disease pathogens to prevention and to treatment of infectious diseases. And so even though we had this high level of confidence that we understand the pathogens, we understand that the way they interact with the host can lead to disease, and we know how to address and prevent that problem, in the recent pandemic, of course, there has been this novel appreciation of a much more in-depth characterization of pathogens that cause these outbreaks and the ability to translate our understanding about these pathogens to effective interventions. And so here you can see how the recent pandemic has really pushed the boundaries of what we know about intervention and treatment, essentially making COVID-19 one of the leading causes of mortality for pulmonary diseases that have been around since, for decades now. And so what that had led to is a greater appreciation of the role that bioinformatics can play in our monitoring, characterization of infectious diseases, the way that they spread, and how we can treat them effectively. And so here you can see a slide from the New England Journal of Medicine, where they describe some of the applications of genomic sequencing to monitor these outbreaks of infectious diseases. Now, one of the first infectious outbreaks that has kind of been a breakthrough moment for genomic sequencing was the outbreak of Ebola virus in West Africa in 2014-2016. During that time, for one of the first times in history, genomic sequencing was applied in on a large scale to monitor and characterize the Ebola virus that was causing that outbreak. But you can see some of the other insights that could be gained from the analysis of that data, including insights into virulence, protein expression to understand potential proteins for drug design, proteins and immune response, diagnostics so that you can really understand the type of a strain or even a specific variant that is causing some particular aspect of the clinical manifestation of disease, insights into growth requirements so that you can understand drug susceptibility, specific sequences for strain detection and outbreak investigation. Potentially there could be zoonotic transmission or a rapid increase in transmission that could be associated with the genomic sequence. So really, genomics has become a central focus for some of the infectious disease monitoring and translational research. And that's why using a lot of these applications combined with the immunology of disease is really so centered around data management, data analysis, and interpretation. And so when we speak about bioinformatics for infectious diseases, some of the examples that we're going to be looking at is how can we compare different sequences from different similar viruses that are nevertheless divergent over time and causing different clinical manifestations? How can we characterize the molecular level of infectious disease so that we can compare the immune responses and the type of responses that the human body has to these pathogens? How can we identify some of the important regions on proteins that are so critical for the infection itself? And how can we incorporate our ability to manage and analyze data to integrate this information together and gain some reliable and interpretable biological insights into disease progression, transmission, and treatment of, specifically, we will probably focus on bacterial, viral, and parasite pathogens that cause some of the major diseases out there, like COVID-19, Ebola virus, malaria, tuberculosis, and others. So before we continue, I want to ask if there are any questions. So again, if you have any questions, please put them in the chat. I'm going to just give it one moment here for anyone to write it out. So the objective of this session today is to really make sure that the topics of the upcoming program are clear, that you understand how you might benefit from joining this program. And if you have any questions, either about the topics that I've covered so far or about any of these follow-up topics, please put those in the chat and I will address them either now, or if I do have something prepared already, I'll mention that as well. Okay, so if you have any questions, you can continue posting them in the chat. I will pause in a few more slides and address them as well. Okay, and so Arjun is asking, is there a certificate of completion? How valuable could a certificate of completion from this course be in application to a master's program? So this is a great question. I think that anyone that is trying to learn bioinformatics, one of the important part of mastering bioinformatics, I think, is to apply it to a project of your own. And likely you have a thesis or a project as a part of your master's. And that's one of the things that you can really focus on during this program. I don't think that people today, when they hire people from master degrees, are looking so much at the certificate that you will get. I think more so they will be looking for how knowledgeable you are to apply what you have learned to tackle a certain problem. And so we'll discuss several case studies in this program that will allow you to see what is the application of bioinformatics from beginning to the end of a research project. So how can you find a good data set? How can you plan your analysis? How can you complete that analysis? And what are some of the important critical questions, research questions, that you can ask about the data set that you have found so that the analysis that you present addresses those questions in a comprehensive way? So you will get a certificate of completion, but again, I think the major outcome for anyone in a master's level will be to complete a project and demonstrate your understanding of that problem. So Arjun, let me know if I answered your question, and I'll be happy to expand on that later on. And then Lewis is asking, can I share the last two publications showed in the slides? Yes, so we will share all of the links for these publications after the session is over. If you could reach out to us by email so that we have your email address, and we will be happy to share that with you. Okay, and what does it take to complete a project? So we'll talk a little bit about the different stages of this program. As I mentioned, it's going to include training sessions and interactive sessions like we're having now. It will include some practical introduction to methods of analysis so that you can take curated data sets, process them, and make sure that you're getting the right results. But also at the end, we will leave some time for you to share with us a proposal on a project that you might find in the public domain. So data sets from NCBI virus, from GIS-Aid, from other types of public domain repositories like maybe MALARIA Gen. So if you know of a data set, you will have plenty of time to find and kind of read the literature about that data set, propose a project that you describe just kind of based on your understanding of the logic. This is the data set, this is the question or a comparison that I will ask. Maybe you will perform some exploratory data analysis, and at the end, you'll have the opportunity to get some feedback on the project. And so you will be able to address any issues that come up. So you can maybe enhance your project, make it more comprehensive, make it more interesting, maybe make it more focused on the topics that are relevant in that process. Okay, so Abukari, let me know if I answer that question. Okay, great. So because we are living in the time of the pandemic, right? We probably all have heard about SARS-CoV-2, the spike protein, the genomic variation of that spike protein. And I think that, to get started with doing something meaningful in this particular pandemic, the first thing that we need to understand is how relevant the different data that is available to an impactful question, right? And so I think that I would divide this training into two parts. The first part, which you might as well do on your own, or you can do it together with our group here, is to learn about some of the basics related to where did this virus come from? How do we use bioinformatics to analyze data and test some of the hypotheses that were proposed? What are some of the basic outcomes of the analysis that we can use to answer some of the questions? And so I'm going to go through a few examples, and this is the first one, and I'm using it as the first one because this kind of an introductory project is something that you can try out after you have completed your registration on this portal. So again, I know that we already did this in the beginning of today's session, but if you have already logged in, let's go back to learn-omicslogic.com. And if you haven't, please register on this portal so that you can follow along and see some of the examples that we will be showing here in the next few slides. Okay, so this is the link. You will see that once you have logged in, you will have a profile, and you will have some personal suggestions based on your progress. So as you start completing some of these tutorials and mastering some of these skills, it will start changing as you go through the coursework. And so right here, you see that we have the COVID-19 origin and pathogenesis of SARS-CoV-2. So I'm going to share that link with you as well. Again, if you haven't registered, registration here is free. So just please go ahead and get started. And one of the first things, and this I think will provide you with a good understanding of how this program will work, you will see setting the background, right? Setting the stage for what kind of questions should we be asking and how to translate those theoretical questions to practical data analysis process. So you'll see here some introduction about the coronaviruses and what do they do, what is the important part of it with transmission, and then you will also see what are some of the important factors that you can learn about here. And here you will see some of the objectives, some of the specific objectives that will be covered in this course, in this project. And so similar to this project, in the course of this whole program, we will actually go through quite a number of examples like this. So this one is on SARS-CoV-2. Another one that we will go through is malaria, where databases like MALARIA Gen and PlasmoDB offer us access to a comprehensive collection of data from different regions where malaria is endemic to identify some important factors on drug resistance, on transmission, as well as vaccine efficacy in application to different types of data about different proteins, different populations, different years. And so we'll be exploring that data together as well as a part of this program. As I mentioned, one of the first examples where pandemics or epidemics have been characterized at the genomic level almost in real time is the Ebola virus. And so one of the projects that we will be focused on is that tracking of Ebola virus evolution across that 2014-2016 epidemic in West Africa. And so as a result of these projects that we will dive into to understand their background, to understand the specific questions that could be asked about them, and understand some of the tools and methodologies, you will be able to appreciate the role of omics data in infectious disease research and the application of bioinformatics to be able to ask specific questions about these data sets. And so that will give you an insight into viral genomics and evolution, immune response biomarkers, and the relationship between microbiome and infection. Now, at the core of all of these types of omics data, we will primarily focus on next generation sequencing. And one of the aspects of next generation sequencing is that the technology itself has evolved rapidly over the past couple of decades, essentially leading us to where we are today, where there's just millions and millions of sequences and hundreds of thousands of data sets that are available for anyone to explore and access in the public domain. That means that as someone that comes without the lab capabilities of generating your own data, you can have access to data sets produced at top labs around the world in the best academic centers where people that have been trained for years are sharing data that they generate, and all of us can just take that data and analyze it. And what that means is that it eliminates the concerns about the quality of data. It eliminates the concerns about the quantity of data. So both quantity is there and quality is there. And so I think that makes this an exciting opportunity. And I think that infectious diseases is one of those areas where data is even more prevalent and available than some of the other conditions that you might be interested in. But to understand this data, we have to appreciate the heterogeneity of this data as well. So essentially, as I mentioned, the sequencing technologies have evolved over the years tremendously. And now we have short read sequences, long read sequences. Some of the technologies are much easier to generate quality data. Some of them have some of these other issues. And so we will review the different types of next generation sequencing data and data types before we jump into that. And so before I go into that process of next generation sequencing, again, I want to ask if you have any questions about what we have covered so far. So what we covered so far is the overview of the Omics Logic Learn platform or portal, where you will have access to data sets and tutorials that you can learn from asynchronously. Number two, we started talking about infectious diseases and how research in infectious diseases can be conducted as a part of this program, where you will be able to apply the tools that you learn to data sets that you find on your own in the public domain. Number three, you will understand the different types of data that are available for the questions that you might be asking. So you really have the freedom to choose the type of a project or the type of an application or a question that you might have about these data sets. So again, if you have a question, please put that in the chat box, and I'll be, if I see the questions now, I'll be answering them now. If not, then we will wait until the next pause that we will have to answer any questions that you might have. Okay, so Dr. Amitabh is asking, is the best pathway to complete the introduction to bioinfo course before enrolling for this one? Yes, if you are completely new to bioinformatics, what I would recommend, and let me actually show you a couple of things where you can get started, depending on kind of where you see yourself going with this process. There's a course on Introduction to Bioinformatics that explains the different applications of bioinformatics to biomedicine, to agriculture, etc. There's also a course called Bytes and Molecules. So Bytes and Molecules is a course for somebody who's just beginning to kind of learn about some of these topics. And you will see that here you will have an introduction to the Human Genome Project. You will see the application of citizen science. How do these two kind of paradigms converge together to create a need for bioinformatics or for training in data science? And then you will see some of the basics. What is the genome? What are nucleotides? What are amino acids? How do we go from sequence to function, right? So some of those basic things are going to be explained in this course as well. So either one, Introduction to Bioinformatics, is kind of probably the next step. And then Bytes and Molecules is the kind of first level where you would begin as a newbie to this topic. Does that answer your question, Dr. Amitabh? All right, excellent. So let's go on. So next generation sequencing. To appreciate the opportunities provided by next generation sequencing, we really need to understand how next generation sequencing works. And the way it works is that there's a machine that essentially takes the raw biological material, or RNA and DNA, and converts it into digital data. And this data consists of short sequences, or reads, as we call them, that now represent a digital snapshot of what happens at the subcellular level. Now, when we speak about infectious diseases, that means that we have millions and millions of reads about that pathogen, the virus, the bacteria, or the parasite, and potentially how does it actually look in a real biological sample. And it looks like this, right? So this is the result of mapping those short reads onto the reference genome. This is, by the way, one of the first examples of mapping the sequencing data from the Wuhan outbreak onto the reference genome of the previously known SARS-1 virus, where you can see how many, many short reads provide us with an understanding of what is the consensus sequence that we're ultimately going to be looking at. And so to be able to analyze this data, we can perform what's called sequence alignment. And the sequence alignment could be applied in two different ways. One of them is alignment to a reference, when we have the raw reads and we want to understand the consensus or the variation from the reference. Or if we have a number of these consensus sequences, how do they relate to each other, or multiple sequence alignment, where we can now explore the relationship between them based on variation that we see in the sequences themselves. And so to see the application of how these methods could be used, let's go on to the server.tbioinfo, where we will see how we can analyze this data. And this is going to be for anyone, even if you don't have any background in bioinformatics at all. So I'm going to go to server.tbio.info. If you registered on the Learn Omics Logic portal, you can use the same password that you used there. Maybe some of you already have a password, so just log in. And we'll take a look at one of the sections to kind of see what the process of analysis consists of. So once you're here, we will see how this portal has several different topics that you could apply analysis to. And that includes analysis of next generation sequencing data, mass spectroscopy, structural biology, as well as methods for data integration and modeling. And so what we want to do is see how analysis of these sequences can help reveal relationship between these sequences in application to a question that we might have about the SARS-CoV-2 virus. So I'm going to go under Phylogenetic Tree, and I'll find here several different demo examples. And one of those examples is going to be this one, SARS-CoV-2 origin, right? So it says I have to click on the dropdown, and then I have to click on demo SARS-CoV-2 origin. So as a demo, as an example, we don't have to upload the data. The data is already here. What we need to do is we need to click on several of these buttons to see how this data could be analyzed. So what is the process? What is the flow or the logic behind such analysis? So we have two options. As we mentioned, we can upload FASTQ or FASTA data. In this case, you can see the input files, and those are bat coronavirus, bat SARS-CoV-2, human SARS-CoV-2, as well as some pangolin data sets on SARS-CoV-2, right? So the question here, and this is the question asked in the tutorial that I shared earlier, is what is the relationship between different sequences? How could they be organized into what's called the phylogenetic tree so that we can analyze their evolutionary closeness, how closely related they are, and when could they have diverged into these separate directions of adaptation? And that could help answer the question, what is this virus in humans, and how could it be related to some of these proposed intermediate hosts like bats or pangolins? So I'll click on Start. Then I'm going to use multiple sequence alignment to compare these. I will convert them to amino acid sequences so I can understand the impact of those mutations on the protein sequences. And then I will use all phylogenetic methods that are listed right here to perform this phylogenetic analysis. Now, once I have many different phylogenetic trees based on different models, mathematical models that kind of represent the molecular clock or the molecular evolution that I observe at the sequence level, I can also analyze the phylogenetic tree structures to identify the best one that is kind of convergent between these different trees that I've gotten as a result of many different methods of analysis. So after I'm done with this pipeline, I will have here several different outputs. And I'm going to take one of these to see whether I can understand something about the relationship of these viral genomes. So when I open this plot, I'm going to see here different names, and these names represent different FASTA files that I have uploaded. And so to appreciate the results, I have to annotate them. So essentially, I have to look at how are these represented as a phylogenetic tree, right? So each branch here represents a split from the origin, and the nodes represent a common potential ancestor. And so we can use this analysis to understand how evolutionary time or adaptation, the process of evolution that we observe at the genomic level, has gotten us the present day species that we have collected, the samples that we have collected. So if we apply this technique to Ebola virus, we can ask the question about how do different regions like Gabon, Liberia, and Great Britain relate to each other based on genomic sequences. If we look at the SARS-CoV-2, we can also identify, as I mentioned, the relationship between the human SARS-CoV-2, the bat coronavirus, RAG13, or the bat SARS-like coronavirus, as well as the pangolin and other types of SARS human viruses. So we can see the relationship overall. We can also find specific amino acid mutations in critical regions. For example, this spike glycoprotein mutation, D to G, that was prevalent and became one of the dominant mutations, potentially linked to increased transmission rate in the SARS coronavirus. So that is the second part of what we will do in this program, and we'll talk about how do we find what are called mutations or SNPs. And so these SNPs or mutations or variants are important to be able to analyze, categorize, and characterize in terms of their impact on the phenotype. And so once we perform alignment, and we can perform alignment on nucleotide and amino acid level, what we're interested in are different types of mutations: missense mutations, nonsense mutations, and frameshift mutations. And we want to find particularly those positions that could indicate a change in the protein structure. So it could be done using multiple sequence alignment, or it could be used by looking at all of the reads that we have that represent potentially many different viruses present in a sample and identifying variants of importance. And that is called variant calling. And so by variant calling, we can identify those SNPs that have a potential impact. And then we can use different methods of characterization of protein function to understand how relevant they might be for a specific question of the impact of that mutation on a structural component that we have identified as important to what we're studying. And so here you can see several different ways that we can look at that, and several different ways that we can interpret this kind of information. So what we'll do in the program itself is we'll actually take these models of proteins that are on the host side. Like for example, here you can see the host side ACE2 protein, which is in blue. But in reality, this protein is actually part of the complex, and this complex sits within the BO81 sodium-dependent neutral amino acid transporter. And then we'll also find the spike protein, which also what we have from crystallography studies is just a component of that protein, but we'll look at how they relate to each other by using UCSF chimera and kind of mapping those 3D structures onto each other, helping us understand how this virus actually binds to the host cell. You can see the membrane right here, and then try to make the interpretation of a mutation, of a change in the structure based on the impact that that would have on protein-protein interaction, just like we're seeing here. So again, this will give us some insight into how spike protein of SARS-CoV-2, which is a critical protein for both the transmission, but also the vaccine, prevention of that transmission and prevention of disease, play a critical role. The mutations, right, those variants play a critical role in all of those aspects as we anticipate this pandemic to continue and spread. And so in this program, the Bioinformatics for Infectious Diseases, we will cover sequence variation, evolutionary relationships and how those are measured, functional inference for sequence to structure and function, as well as host response. How can we analyze the response of the host, transcriptomic data, RNA-seq data, to find patterns that can explain some of the reactions that we see between mild, maybe severe disease, maybe asymptomatic disease, right? Maybe we can look into the immunology of different immune cells, etc. Okay, so any questions about the topics that we will cover in this program? Again, if you have the questions, please put those questions into the chat box and we will be answering those. But for the sake of time, I am going to now pass the session over to Dr. Mohit Mazumdar. So Mohit Mazumdar has been working with us to organize the schedules and the logistics of many of the programs and adapt them to the needs of different levels of participants. So what he will do is explain a little bit more about the different sessions and the topics that will be covered and how to register for this upcoming program. So Mohit, over to you.
Thank you, Ilya. Okay, let me share my screen. Hello, everyone. My name is Mohit Mazumdar, and I have 12 years of experience working and collaborating with pharma and healthcare industry to solve high priority scientific questions related to biomedical data science and bioinformatics capacity building, as Ilya mentioned. So some of the things that I would want to mention before we move on to the program overview is that the pace at which firms have, like at this current time, the pace at which firms are collecting and processing real-time data is simply astonishing. So I would wanted to give you some examples, like tech firms in Asia have developed mobile apps to help people check if they have taken the same flight or train to confirm the virus patients. Then government officials are using data to track individuals that uses the same data. And many apps are there which uses infrared and AI-powered facial recognition to screen people at using this sort of data. At the same time, Google's DeepMind is working to find out about the structures of proteins that are associated with COVID-19. So I think a lot of things are happening with data, and for this generation of researchers and students, there is an opportunity to discover from big data and contribute. So in this specialization infectious diseases program, we will be using curated data sets on infectious diseases such as malaria, Ebola, enterovirus, COVID-19, as I said, coronavirus, host response data, and we will be using public repositories such as NCBI virus. So in this program, we will understand about the viral diversity, and we will learn about bioinformatics analysis approaches to study about the viral genome. And also we will gain hands-on experience throughout the sessions that I'll be showing you next. So in these sessions, how will we be using and analyzing curated data sets, for which we have already prepared those example projects for you to look at and gain that experience. And then on top of it, you will be guided by experts with bioinformatics experience and knowledge about virology. So the first session is scheduled for 18th May. So we are going to start the session at the same time. So the first session will be about next generation sequencing and viral genomes in host transcriptome. And then after that, the next session. So we have 10 scheduled sessions, which will be happening over a period of a month. So the first session is on 18th May, the second session is on 20th May, and we will be sharing you the detailed schedule of the program via email as well, and it's also there on our website. So if you want, you can look into that after the session as well. So the second session, as I was saying, would be about multiple sequence alignment and phylogenetic analysis, and this will be followed up by a hands-on session about preparing and running your own pipeline. So we will be doing multiple sequence alignment of viral genomes, and then we will be building phylogenetic trees, and then finding full genome sequences and preparing those FASTA files. So all of that will be covered in this third session on 25th of May. And then we have a Q&A and discussion of pipeline results. So all of the data that we will be generating and the results that we'll be getting, we will be analyzing those and understand the biological significance or the significance in the context of the research. So in that sense, we will be using and learning about FASTA and FASTQ files, which databases to use for detection of viral genomes by mapping on those genomes to databases, and then interpretation of phylogenetic interpretation, where we are talking about evolutionary relationships between genomes and all of that. So after that, the next session is about the infection to pandemic, so the viral adaptation. Here we will be talking about hosts and origins. So this will be on 1st June 2021, and transmission, and then cell entry and tissue tropism. After that, on 3rd June, we have this session, which is about the symptom severity. So here we will be talking about the disease progression and the outcomes of that, how the virus is replicating, and what is the process of immune invasion, and how we can see that through the data. Followed by that, we have a hands-on session again on 8th June. So on 8th June, we will be working on the origin of human infection with taking examples, and this will be a comparative study because we'll be looking at different viruses like MERS, SARS, and SARS-2, and comparing their origin by looking at doing the sequence analysis. So this will be on 8th June, as I said, and this will be followed by the next session, which will be on 10th June, so which is about the variants. So the rate of mutation and how to identify variants, what are the point mutations, substitutions, insertions, and about the mutations, like what are the synonymous mutations, what are the non-synonymous mutations that you're hearing about so much, and also like the current scenario with COVID-19 and the variant analysis that is also a part of this program. So that will be done on 10th. And after that, we have the session on annotation and significance of the analysis. As I was keep on saying, that relating the results that we get from big data, and then connecting it to the biology or to the analysis results. So we'll be looking at the location on genome and protein function. So that would be on 15th June. And the same week, the next session will be about host pathogen interaction, where we will be discussing about the protein-protein interaction that Ilya discussed, and the host response and the immune response. So what is happening with the adaptive and innate immunity, and then about that, considering the bioinformatics and the holistic understanding of the system biology, considering all what we have learned, how this can be factored in for vaccine design, how these all plays a role with a data-driven decision to go for a very specialized sort of vaccine design that could address the problem of the variants, that could address the problems of the changing epitopes and all of that. So that would be on 17th June. So that would be about the schedule. And I'll pause here for a second, and I'll look at if you have any questions about these. So if there are no questions here, okay. Okay, so
again, I just wanted to mention that the recording of this will be made available on our YouTube channel, and we'll send a follow-up email to those of you that asked any questions. So some of you asked for some of the reference publications or how to register. So that will be shared by email as a follow-up.
Yeah, okay, thank you, Ilya. And then, as I was saying, that once we have learned about this, the next opportunity, as we've been discussing, is to be able to come up with a research proposal or to be able to work on a project. So this independent project or research proposal is the time when we develop within that period of 1 month, by talking to you, by one-on-one meetings or in a group meeting, and discuss that how we can leverage the online publicly available data sets and the tools that we have available here, the cloud infrastructure, and to come up with research proposals that could be new research. So that would be about the program. So this program will consist of several resources, and some of the resources, as Ilya have already shown. So one of the places that you should go to is the Learn platform. So we have in this entire Omics Logic program have has several components. So those components include the training material, data analysis tools, curated project examples, and interactive sessions such as this. So I hope that that part is clear to everyone, that we will be using multiple resources to come up and learn about this innovative and new field of bioinformatics for infectious diseases. So I think if you have not yet created a login on the Learn platform, so please do that. So it's a free login that you can create and use some of the free resources that are there, and then you can also choose to join the subscriptions that I'll be showing you the next. So this program will be utilized. You will also utilize the TBioInfo platform, where we will be running the analysis, big data analysis, and there are several sections, as you have already seen. Many of you have maybe have seen the demonstration. Use the demonstration passwords and email by your own email. So yeah, obviously you can do that as well. So also after that, we will be utilizing the coding platform as discussed, and we'll be utilizing some of the analysis, downstream analysis that are required for us to learn from this data. So this would include the visualization and getting classification done for this for different types of taxas. So out of this entire experience, you will be able to understand from the scientific literature and that describes the omics data. So you'll be able to perform next generation sequencing analysis with an accuracy and with reproducibility, and you'll be able to do exploratory data analysis and generate an hypothesis. So that is something that we discussed for the projects, and then do statistical analysis and do some of the machine learning analysis if required for you to go into the omics data and find out those patterns. And then we do the annotation using the databases that are available online, and then we find out the relationships and write about a story and write about what we have found in our entire research project or research program. So that is something that it will encompass. And other than that, I would like to now take you to the registration page, which is fairly simple. So just a second. So let me share you with the registration link, and or Ilya, if you can share the registration link.
Actually, I'm not sure what the registration link that you'll be using, so please share.
Just a second. So we have already emailed the registration details. So in case if you have any questions, feel free to put them in the chat and we'll be able to answer that. Meanwhile, let me share my screen and show you the registration process. So this is the portal, infectiousdiseases.omicslogic.com. So let me put it in the chat box. And to answer the question about Shayani about the course fee, so I'll just be showing you that. So here you can click on get started, and then here you can choose from 3 different levels. So beginners, intermediate, and advanced. So here you will be able to get the information about different levels. So the beginner level has all the components that we have discussed today, including all the licenses that are there and all the platform access and all the projects. And the difference between intermediate and beginners is that you will get an additional mentor support in the intermediate package, and in the advanced one, if you're working on your own data, if you're working in a lab that is generating data and you would want to analyze that, which is not available on public repositories, then you can choose for the advanced license and collaborate with us to work on your research project. So anyways, to tell you more about the pricing, the cost of this beginner's license 1 month hands-on program is $205. And we have a 30% discount code available on top of it. So you can select here and you can write the 30% discount code. And with that, that would be the price or the cost of this program, provided that you get the early bird scholarship. So these are 3 different methods that you can pay through. And you can also pay via bank transfer or UPI if you are if you don't have a card, or if you want that help and assistance for joining the program. So please let me know if you have any questions or any clarifications that are required for you to understand about this